a lot about how I wanted to introduce my dissertation talk to you today. If you've been around for a bit, you've seen me give a variety of office clips or nails on a chalkboard or any of that. I'm not going to torture you with that today. Instead, I'm going to actually show you the video that started it for me. So this is a video from ABC News 2020, a special that occurred in May of 2012. So this was back when I was in high school. And this is the special that first told me that misophonia existed. So I want to just show you a couple short clips from this, and we will see if it works.
And we start with something.
Nope.
Wait, unmute your.
I'm unmuting.
Something we've all felt.
Nope. Hold on. Oh no. That's okay.
And we start with something we've all felt at least a little bit. Those pet peeves and annoying little sounds our loved ones might make. But for the people you're about to meet, take that and pump up the volume a lot, because they have a condition so rare most doctors have never even heard of it, even though it's all about hearing. 14-year-old Taylor and her mother Bernadette eat meals in separate rooms. They share no activities. In fact, they do not even speak. No normal things, because Taylor suffers from a mysterious condition called misophonia. It means hatred of sound. For Taylor, everyday noises like chewing, coughing, and sniffling trigger emotions that bring her to the brink.
It's an almost undescribable amount of anger and rage that I just can't control.
In fact, the condition has received so little research, most doctors have never even heard of it.
So this was cool to me. Oh, nope. Testing. Cool. So when I first saw this back in 2012, I was like, holy cow, this thing that I'm experiencing has a name. But as you saw in this clip, most doctors had never heard of it. What is this rare condition? And I'm stoked to tell you today about some research that I've done on this now. And I'm going to talk you through some behavioral, cognitive, and neural experiments I've done to try to understand this rare condition. So first off, also, I do not like that this screen didn't exist. Can get rid of this? Can you go back to mine? I don't know.
Go to bar. I think. I have floating meeting as well. Yep.
Hey, I have a wonderful border that I need you to see the entire time. Okay.
You need to click the PowerPoint again.
Oh my gosh. I've been on Zoom for so long, and I still don't know. Okay, we're here. So starting off with, what is misophonia? So as we saw, as the definition in that video showed, misophonia translates literally to the hatred of sound. But it's a little bit of a misnomer, because it's not a hatred of like all sound in general. It's a hatred of really specific certain sounds. You see some of them depicted here. So maybe like slurping through a straw or chewing, walking in heels, for instance, those types of sounds. Now, we've come a long way since that 2020 clip. We now have what is considered a consensus definition of misophonia that came out literally just last year, describing misophonia as a disorder of decreased tolerance to specific sounds. They call them triggers. And these cause an unpleasant feeling not seen in most other people. Now, the 2020 clip considered it a very rare kind of thing, but actually it's prevalent in about 20% of the general population. And it's such an impairing kind of condition that about 1 in 5 sufferers have reported thoughts of suicide because of their misophonia. So it definitely impacts your life pretty seriously, but we don't really know much about it. In fact, there's not even an official diagnostic criteria to determine misophonia. However, this was back before the definition happened. Some researchers down in Amsterdam tried. So they said that in order to classify as having misophonia, there needs to be the presence or anticipation of a sound produced by a human being, like eating or breathing sounds, that causes this reaction. And this really stuck out to me, both from my personal experience and what I've read of others, because it kind of emphasizes this eating component. And in fact, this criterion has been updated to mandate that oral or nasal sounds must be a trigger in order to classify as misophonia. But I sat there and I was like, is this diagnostic criterion sufficient to really capture what misophonia is, or more specifically, do sounds need to be human produced or from oral or nasal origins to be bothersome? So this motivates my first project. Specifically, I'm asking the questions, are non-human or non-oral nasal sounds more aversive in misophonia compared to controls? And secondly, can I actually predict misophonia severity using ratings from different sources? So let's start with this first question. To answer this question, I chose sounds from 3 different source categories of sound production. So source 1 was that one from the criterion. These are human-produced sounds that are oral or nasal. These are from your nose, like sniffling, or from your throat, mouth, like slurping or chewing. Now, to test against this source, first I removed oral nasal from the equation. So these are human-produced sounds that are not oral or nasal, things you could still blame on a human, like a hand clicking a pen, typing, that kind of thing, but not from the mouth. And lastly, I removed human from the equation, and these are non-human or nature sounds, just to compare exactly what is going on. For each of these 3 sources, I chose 10 different sounds, and you can see them depicted here. So all participants were shown all 30 of these sounds. And the way this worked is they logged in to an online Qualtrics experiment. If you've ever used Qualtrics, it's not made for experiment. But I did this before the pandemic, before online experiments were cool. And I had participants log in. They listened to all 30 sounds one at a time, all mixed together. And at the end, after each sound, I asked them how much discomfort they felt during that sound on a scale from none at all to an extreme amount. After they listened to all 30, they were given some short surveys where I gave them some assessments that exist at the time to try to get at misophonia. So this is my sample. I initially had to do this with self-diagnosed misophonics. So these are individuals who are aware of the condition, so much so that they joined either a Facebook support group or the Reddit subreddit. You can see here what their misophonia levels kind of are on a scale from none to extreme. And I contrasted this with 39 individuals from the general population. And I want you to notice here that some of these individuals actually overlap into the red area, showing that it is present in the general population. Now, getting through some results. The first question I asked are non-human or non-oral nasal sounds more aversive in misophonia compared to controls? So what I'm going to show you here is the 3 source categories on the x-axis and the mean discomfort rating on the y. Now, unsurprisingly, oral or nasal sounds do show where misophonia individuals do have higher discomfort ratings compared to controls. That's not surprising, given the criterion. Here's the real question, though. If misophonia was just oral nasal, I shouldn't see any difference in these latter 2 categories. But in fact, I do. So even though misophonia does involve more aversion to oral nasal, these do also show a significant difference between groups. So I can say that yes, non-human and non-oral sounds are more aversive. So secondly, can I predict severity? Now to do this, I expanded my sound bank. So instead of 30 sounds, now I have 125, still split between these 3 sources. Similar, I had a new group of subjects that you can see here. Again, misophonia and general population. And I'm going to show you 2 different predictions, because there are a variety of different ways you can predict misophonia. The first way, I'm going to ask, can I predict their misophonia level given their discomfort ratings to sounds from each source category separately? And here's how I'm going to do that. I'm going to take my entire sample of participants, and I'm going to equally subdivide them into a training set and a test set, making sure that there are equal amounts of misophonia and general population in each group. I'm then going to build 3 models. Each model is only going to include predictors or the discomfort rating to sounds from that particular source. So for instance, the first model is going to contain 28 predictors representing the 28 discomfort ratings that were for oral sounds. I'm going to construct that model by feeding it discomfort ratings for the corresponding predictors, as well as the misophonia levels of the training set. And then I'm going to apply that model to the left out participants of the test set, feeding it discomfort ratings and predicting what the model thinks their misophonia level is. And it's the results of this that I'm going to show you on the next slide. So I'm going to show you actual misophonia levels on the x-axis and predicted misophonia levels on the y, separately for these three sources. Unsurprisingly, oral or nasal aversion does predict misophonia pretty well. Again, these are the critical comparisons. And in fact, using just source 2 sounds or just non-human nature sounds, I can also significantly predict misophonia. But I thought I'd take this one step further, and I asked, all right, well, which sounds are most influential to determine misophonia? Is it mainly oral nasal, or is it a combination? And to do this, I did a different style of predictions. I did what's called a leave one out cross-validated procedure, where I took all of my participants and I left one out of the equation, and I trained this model on all -1 participants. I used a stepwise regression here, which is going to add or subtract sounds until it reaches the one that most significantly predicts misophonia level. It's only going to keep the predictors that are most significant in predicting misophonia. Once I have that model, I'm going to apply it to the left out person and predict what their misophonia level might be. And then I'm going to iterate through this entire process until every participant's been left out once. That gives me a predicted level for every person. And again, that's what I'm going to show you on the next slide. So here, when I use this procedure, I again can significantly predict misophonia level. But perhaps what was more interesting to me is what sounds got kept in the models. Now, granted, each model was slightly different because they were constructed on slightly different participants. However, there were some commonalities. In 80% of the models, we saw sounds spanning all three sources. So this tells us that it's not just oral and nasal sounds that are the most influential in helping you determine misophonia. You do better if you incorporate all three sources. So yes, I can predict misophonia using all three sources separately, and my most predictive sounds vary in source. So ultimately, I asked this question, is this criterion sufficient? And I argue, no, it is not. In fact, misophonia conception should include more than just oral or nasal sounds, which I think is what led to that definition you saw at the beginning that is more inclusive of all types of sounds. But there's another criterion in this list that I also kind of want to look at. This criterion touches on the impairment associated with misophonia. It says it must make it difficult for the person to perform important tasks at work, meet new friends, attend classes, or interact with others. You saw this in the 2020 news clip a lot. She couldn't interact with her mom. Now, this hasn't been tested as much, but let me tell you a few studies that have happened. In one study, a group of researchers had participants study a written paragraph that they were then tested on their memory for later. But the trick was, one of those individuals was actually part of the research team, and they chewed gum the entire time to try to mimic that sort of triggering sound. They showed in their results that participants with low misophonia or controls had no difference in their comprehension on that memory test if there was gum chewing, a sound present, or a control, no sound present. But individuals with high misophonia show a large decrease in comprehension when they're bothered by those triggering noises. Another study more recently has used the Stroop paradigm. So this is when a colored word is written in a color that is either congruent or incongruent with its identity. And it typically, you'll see that it takes you longer to produce the color of the sound when those two things are incongruent compared to when they're congruent. So they had participants do this Stroop paradigm while listening to oral or nasal sounds. And they showed, comparing the Stroop effect, so how many seconds delay that takes you to say that incongruent color, compared to misophonia level, shows a small but still significant correlation. So the higher misophonia you have, the slower you are at producing that response when you're in that bothersome environment. Now, there's one other cognitive thing that I want to tell you about before I get into this experiment. And that is actually a thing that I found in the supplemental section of my first experiment. So for some of those studies, I asked participants to identify the sound they heard for me. And they wrote their answer into this little text box. Now, I got some really cool kind of social insight into misophonia. For instance, I had participants tell me, it sounds like my brother chewing. Or they assign these kind of negative characteristics, like it's a crunching cave person with no manners. But my favorite is this person who wrote me an entire paragraph into this tiny little box, who said, at first, I thought it was sliding an object around on a desk, which when I thought it was that, it didn't bother me. Then I thought it was sniffling, and that did bother me. And this was really fascinating to me, because this demonstrates that the same acoustic sound can bother you in some cases and not in others. And so I looked at my data and I looked at the affective identity and found that when participants with misophonia correctly identified an oral or nasal sound, they in fact rated it as significantly higher discomfort than when they got it wrong. But this isn't just a universal phenomenon. It wasn't true for those non-human or nature sounds. So I wanted to test this more officially. So for this cognitive experiment, I'm going to ask these specific questions. First, does the ability to identify a trigger sound influence how aversive it is perceived to be? And two, does the presence of these trigger sounds impact your social or cognitive processing? So to answer these questions, I sampled from 26 individuals with misophonia and 39 controls from the population. And I presented them different categories of sounds. So similar to before, I have an oral category. You see here breathing, chewing, etc. I have a non-oral human category, cutting food, dribbling, etc. I also have control sounds. So these are sounds not meant to be bothersome. It could be just ambiance, or like a white noise hair dryer, or pleasant like a harp. And I'm contrasting this with no sound at all, just to get the effect of auditory stimulus in general. Now, these again are the 12 different categories I have. And these sounds came from an open access misophonia sound bank that I and collaborators have constructed. The cool thing about the sound bank is that it incorporates multiple exemplars of each of these different sounds. So instead of just presenting one instance of breathing in my experiment, which you can see could look very different depending on the clip I use, I am showing 4 different types. So this helps to kind of ensure that a person who is bothered by breathing is bothered by at least something in this study. And it's a little bit more representative of the real world. Now, the way this worked is I had a 2-phase experiment. In the first phase, they learned faces. So the way I had this was that while a sound was playing in the background, participants viewed a face, neutral in posture, and their task was just to determine what gender the face most presented as. After the sound was over, they then gave a rating of both what sound they had just heard using a 12 alternative force choice and their anxiety or discomfort on a scale from 0 to 5. After they listened to all of those trials and sounds, I then asked them to give me a likable rating of the same faces they just saw, intermixed with brand new novel faces. So they told me how likable they found that person on a scale from 1 to 7. They then gave me a memory judgment of whether or not that face was old or new. And then lastly, they gave me a sound memory judgment. If that face was old, what sound initially was paired with that face? So a lot to look at here. Let's start with this first question. Does the ability to identify a trigger sound influence how aversive it is perceived to be? To answer this question, I'm going to use that sound identification and discomfort rating from the first part. So here, if I show you sound identification on the x-axis, correct versus incorrect, and mean discomfort on the y, I can show you that again, oral and non-oral triggers do show higher discomfort when the sound was correctly identified compared to when it was incorrectly identified. But again, not a universal statement, because it's not true for control sounds or quiet sounds. So there's something about triggers needing to be identified. So yes, I showed in this more directly that sounds are more aversive when they're correctly identified. So secondly, let's talk about some of that impairment. First, does it impair or impact social processing? And to answer this question, I'm going to look at that likable rating that I had participants give. And spoiler alert, when I split my sounds by my predetermined categories, I don't get any difference in how likable they rated these faces. I have a slight marginal affective group here, misophonia being less than controls, but not significant. So instead, I looked at this and I said, instead of my predefined category groupings, there are individual differences in how people find these sounds. So instead, what if I take each participant and I rank order the sounds that they found as most uncomfortable? And I can call then their top sounds high discomfort sounds, and their bottom sounds low discomfort sounds. So what if I analyze likable ratings instead, using these categories instead of my predetermined ones? So here, if I look at how uncomfortable they found the sound that was initially paired with the face, I see that indeed, participants do find faces less likable when the sound that was paired with them they found as high discomfort. But I can look at this one more way, and that's also what sound they remembered being paired with that face. So if I look at it this way, what sound they reported to be present, I see kind of cool findings. So when a participant reports a high discomfort sound, they in fact see that person as less likable. And this is even more so true for individuals with misophonia compared to controls. So if someone with misophonia reports their memory as being a sound they do not like, that person was less likable. So let's look at cognitive processing. And I'm going to answer this question in two different ways. The first is cognitive processing during the task. So while the sound was playing, that gender judgment. Initially, this was just meant as an encoding task, but I actually found some pretty interesting results. So when I look at both the accuracy of that judgment and the response time of that judgment, I see that participants are in fact slightly or significantly less accurate when the sound was an oral or nasal kind of sound, and also when that sound was a non-oral human sound compared to control or quiet. And additionally, they were slower to respond on that judgment when it was a trigger sound compared to when it was control. So this shows me that they are impacted even just in the moment to complete a task as simple as gender judgment. And lastly, I can look at their memory for that face. So if I look at face accuracy here, I can see that participants were less accurate at identifying the face when it was paired with the trigger sound. And if I then rearrange my analyses and look at it again by discomfort instead, I see that based off the discomfort initially paired with the face, participants likewise were significantly worse at remembering the face when it was paired with a high discomfort sound. So overall, results are a little bit nuanced here, but I've shown that it does impact social processing such that when a participant does not like a face, they're kind of attributing it to a sound they don't like. And secondly, it impacts cognitive processing, both decreased performance during that gender judgment and worse memory after, although this seems to be an effect not specific to misophonia in general. Now, I've shown you that misophonia does have some measurable impacts, but can we ascertain what even causes misophonia? So to answer this question, we're going to look at the brain. And I'm going to tell you of one study in particular that first came out looking at the brain. They said, all right, since most trigger sounds are generated by the mouth or nose, orofacial movements, we hypothesized that the mirror neuron system related to orofacial movements would underlie misophonia. And just as a reminder, the mirror neuron idea is this idea that we first found in monkeys that showed that we have neurons in our brain that are active both when you yourself do an action and also when you watch someone else do an action. These are neurons that you find usually from like single cell recording or in electrodes. But to answer this question, these researchers decided to use resting state connectivity instead. So what this is measuring is the fluctuation over time of different regions of your brain, showing that brain regions that tend to fluctuate at the same time with each other, we consider to be functionally connected, and regions that are more disparate are considered to not be connected. So they're looking at brain connectivity over time. They're looking at connectivity from different areas of the brain, specifically auditory cortex, which we know is in the temporal lobe and does things as low level as frequency information, and as high level as speech understanding or kind of what the semantic content of speech and sound is. They're also looking at the insula, this little part inside of your brain that does a variety of things, from subjective response to pain, to disgust, kind of emotion regulation, to attention and cognitive control. And lastly, they're looking at the motor cortex. This is kind of their main meat of their finding. So we know that we have this strip in the frontal lobe that has a representation for every part of your body, and it's in control of kind of moving that body part. So they're looking specifically at this mouth and face kind of region of the motor cortex, which they specifically define by using the part of motor cortex that showed stronger connectivity to auditory cortex in resting state, which is a little bit maybe roundabout in how I might go about defining that region. But let's see what they found. They showed misophonia had significant increase in connectivity between those same regions, auditory cortex and orofacial motor cortex, which again is how they defined it. So a little bit of a red flag to me. And lastly, they showed that orofacial motor cortex had increased connectivity with anterior insula and misophonia, which is an interesting finding given how function of the insula does relate to misophonia. They concluded overall, misophonia is characterized by increased resting state functional connectivity between orofacial motor area and auditory cortex, which again, a little bit of a red flag because that's how they defined the orofacial region. And in case you were like, oh, well, no way that they're actually going to make huge claims with that, they say our data strongly support the hypothesis that misophonia is characterized by aberrant functioning of the mirror neuron system, which again can only be found with single cell recording, not resting state connectivity, which they admit. They say demonstration of mirror neurons requires invasive recording. fMRI can't do that. But they still run with this conclusion. And since I'm particularly interested in these non-oral nasal triggers, they addressed that briefly. And they said a plausible explanation is that after typical triggers are learned via the mirror system, non-typical stimuli become triggers via some other mechanism later, associative learning. So I looked at this and I was like, all right, I got to try this myself. So my first question is, can I replicate these findings? But instead of using a resting state method to determine orofacial, I'm going to use a functional region of interest to find using a task. And secondly, because I'm interested in these non-oral nasal sounds, is there any neural evidence for mesophonic aversion to non-orofacial stimuli? So to do this, I had 19 adults from the general population that I separated post hoc into higher and lower misophonia groups. My method was similar to the prior study. So I had a resting state scan. This is when a participant is just laying in the scanner without any particular task to focus on. So they're just seeing what naturally is going on in their brain. I also gave them a task, which I'm going to pin for a second and come back to. I have similar regions of interest as before, auditory cortex, insula, motor strip, but in addition, we have an analogous region, an analogous strip in the sensory area that is involved in the sensation of your body parts. So I'm going to test the specificity of the motor hypothesis by comparing it against sensory information too. Now, the key kind of difference is how to define where orofacial cortex is. So I'm going to do this two different ways. First, I'm going to model what the previous paper did by saying whatever voxels in this mass are most strongly connected to auditory cortex, I'm going to call those voxels orofacial cortex. So this is modeled after the previous paper. I'm going to refer to these as resting state regions of interest, or RS, ROS. Secondly, I'm going to use a task. So this is an articulatory localizer where participants alternated between blocks of phoneme production, so physically saying in the scanner, ba, ga, ra, da, moving your lips and mouth and throat and all of that, contrasted with finger tapping. So this is them tapping their fingers in order, one at a time, over and over, getting finger movement. So by looking at this data, I can define in each individual subject where their orofacial region is that you see in red compared to where their finger region is in blue. So therefore, I can define this cortex more precisely using task contrast. All right, so putting that information together, These are the different methods I'm going to show you in this experiment. So first, using that resting state method, I have two different ROIs, a motor one and a sensory one. And I'm going to look at connectivity between auditory cortex and insula. And I'm going to do that again using my functional method. But now I have both orofacial ROIs and finger ROIs. There's a lot going on here. So let's go step by step. We're going to start with just this top half. And I'm going to answer the question, can I replicate the results of the previous paper? So here again, they found increased connectivity between auditory cortex and this orofacial region. Yeah, and I in fact do show that same exact finding. So yay, I can replicate that previous result in my sample. But maybe a perhaps more interesting question is it motor specific? Do I see any sensory differences in connectivity? And again, I do. So that means to me that it is not just a motor hypothesis that is driving misophonia, but there might be some kind of sensory input as well. So that raises the question, all right, so I'm showing this increased connectivity with orofacial areas. Is that area really responsive to orofacial movement? So to answer this question, I'm going to turn to my functionally defined orofacial area to see. So if what that resting state method did was capturing orofacial cortex, I should see the same pattern of results here that I saw before. But I don't. So that tells me that the voxels most connected to auditory cortex aren't actually responsive to orofacial movement or mouth movement at all. So I can look at this one more way just to test, because again, I have task-based data on each of these people. So I can say, all right, well, what about in that RS region, that RS ROI, are those voxels at all responsive to mouth movement? If I look at my functional method, just as like a sanity check, yes, the regions that I define functionally do show responsiveness to oral or nasal action in an independent run of the task. So that's good to go. But that RS method doesn't. So in these voxels, there's no selectivity to red to that orofacial movement at all. So I've shown here that orofacial cortex isn't selective for mouth movement. So we were able to replicate their resting state findings when we do it their way, but importantly, not when we use functionally defined regions. So lastly, is there any evidence at all for non-orofacial stimuli aversion? And for this, I'm going to look at that last comparison here, that finger region. So if misophonia really was this kind of oral or nasal aversion, and the finger, the non-oral triggers came online later, I shouldn't really see any difference here. There'd be no reason to have brain differences between a finger region in your brain. However, I do see a significant difference between the motor area and insula in misophonia, which demonstrates to me that given this finding, given that there is actual measurable connectivity differences, it suggests there might be a similar neural basis for these non-oral nasal triggers. It's not just some later process that comes online. So overall, I'm showing that there are non-orofacial regions showing differential brain connectivity in misophonia. So let me wrap up. So overall, I showed you a couple different things. First, and primarily, I showed you misophonia is not merely a disorder of oral or nasal aversion. And I showed you this in a couple different analyses. First, behaviorally, that misophonic individuals rate these non-oral, nasal, and non-human sounds as more aversive than controls do. And that predictions are most effective with a variety of sounds. I also showed you cognitively that non-oral nasal sounds do also impair task performance. And neurally, we have evidence of abnormal connectivity with non-orofacial regions. Secondly, I've learned that misophonia aversion is at least partially dependent upon sound identification. So I showed you this in my supplement of the first experiment, that they're rating trigger sounds as less aversive when they didn't know what the sound was. And again, more formally in my second cognitive experiment, where they're rating trigger sounds again as more aversive when they knew what it was. And lastly, and maybe importantly here, I'm showing evidence against this groundbreaking mirror neuron hypothesis. I demonstrated that orofacial region in their paper was unlikely to actually be an orofacial region. I showed that there were also differences in sensory cortex, showing that it wasn't just specific to a motor hypothesis. And lastly, that there was connectivity differences with finger regions as well, showing that orofacial was too specific. But I must kind of touch on a few limitations here. So one important one is that since misophonia is not actually diagnosable yet with some formal criterion, all of these were determined via self-report of assessments that were available to me at the time of the study. And secondly, since there are large individual differences in what people with misophonia find bothersome, my sound stimuli may not have covered all available trigger stimuli. And in fact, my neuroimaging procedure didn't actually present sounds. It was just looking at what your brain is doing naturally. And then my last thing I'm going to touch on briefly is some future directions. So one, there's so much to unpack. We're nowhere near the all we need to know about misophonia. I'm curious about why and how certain sounds become triggers and not others, and what can be done preemptively both to avoid new triggers, but also therapeutically to minimize their impact. And lastly, as Andy was alluding to, I'm excited to move up to Montreal. I'm going to join Concordia University to develop a large open access neuroimaging database of misophonia brains using both MEG and MRI data. And if you'll have me for a minute more, there are things that I need to acknowledge here. So first, I've been blessed with a variety of different funding throughout my time in grad school. Things like the Gibson Award that allowed me to do neuroimaging work, department funds that allowed me to get skin conductance equipment, which I didn't even mention in this talk, as well as some external grants that have allowed me to work on these projects without having to do any sort of external job or teaching in the meantime. As Andy was alluding to, I've gotten so many cool emails that have motivated me to keep this project going. And this one that I'm highlighting in particular, the girl from that 2020 video sent me an email a few years ago, thanking me for doing this stuff and saying I'm the kind of person who will make a difference in this world, which is one of those things that when you're having a troubling day in grad school is such a good mood booster. I also can't go without acknowledging the two labs that I'm a part of, and specifically Zainab and Andy. Y'all have been amazing in this experience. I still remember our interviewing days where you're like, okay, but what if misophonia doesn't pan out though? Like, what if we have you do it this way instead? And like, it made super sense at the time, but like just in retrospect now, it's amazing how much we've accomplished in these 6 years. I also just want to shout out to all my friends in the psychology department and all just the fun that we've had amongst the 6 years. I really enjoyed my time in grad school. And then lastly, my friends and family. Sorry, I left home, but my parents, my brother and sister-in-law, my best friend from home, and also obviously my fiance, Charlie. I'm excited to marry you next week. And so with that, I am done. Thank you for listening.
Wonderful. We have just a quick format here. Excuse me. We'll have a few minutes for questions from the general audience. And then we will pick you out. Even though I said something, I said and I'm confident and excitement for my students. We still have to do an exam so that Heather will be able to be evaluated for the PhD. So that's real. That will happen afterwards. And we will conclude that by 2:30 p.m. So look out for an email to get the actual result, which I'm truly predicting in the future. But, so we have a few minutes for questions. And yeah, please, out the way.
Blair.
This is something I should know the answer to just from knowing you, but are triggers always repetitive, or do people react just to single exposure to a sound?
Are triggers always repetitive, or do individuals react to a single exposure? This is something that I actually wanted to explicitly tease apart in my first year of grad school, to study the repetition, whether or not you needed that bout of silence in between to find it bothersome. I don't know the direct answer to that question. I would say that it needs to be at least long enough that you understand what the sound is and you can kind of respond to it. I've had anecdotally participants tell me that, oh, I knew that sound was going to end in 2 seconds, so it didn't bother me as much. So I think there is definitely something to be said about the duration of the sound and that kind of repetitive nature to it. Yeah.
Fascinating talk. Thanks very much. Good luck on the exam. I have 2 questions. One is triggered by your friends that about the diagnostic criteria on oral nasal nature versus more general nature. It is sufficient or not?
Yeah.
That word sufficient triggered certain things in my philosophical.
Sure.
If you talk about sufficiency, you must find an example in which there is a case of misophonia which is triggered by non-nasal sounds only.
Yep.
But is not triggered by.
Yep.
Only.
Yep.
So do these people exist?
Anecdotally, yes. So I have a friend, for instance, my collaborator in Montreal, totally fine. Everything sniffling, oral, totally fine. She hates beeping sounds and like cutlery. Like there are certain things where, although rare, like I will admit that most participants do at least have something oral that bothers them. I think there are cases where that is not always the case. Yeah.
And that leads to my second question. Yes, that bothers them, but then you mentioned the prevailing emotion is one of anger. And that's, I'm speculating, I would not, I have no data, but is it possible that the chain of events is you hear somebody sniffling and then you start, oh, that person is spreading disease. It's such an irresponsible behavior. This is what makes me angry. So use these mental models. These sounds are not bothersome because they're sounds.
Yeah.
They're bothersome because they're signs of deeper bothersome things.
Sure. I have two responses to that. One, I didn't even touch on this, but other researchers have challenged anger as the main emotion. So other research has shown that it's actually more of an anxious feeling or worrisome than it is an actual rage. So that's like one kind of component. I don't think anger in particular is the sole primary emotion. But two, I think that
rage is super anger, right?
Sure, yeah. But my second kind of response to that is that might explain some of the oral, some of the like health related, sniffling types of sounds, but that doesn't fully cover the picture of like clicking a pen, for instance. Like why would I feel anger that there's no like health reason that I would feel anger toward pen clicking or typing, for instance. So it is an interesting hypothesis, but one that I don't think covers misophonia as a whole. Yeah, Jay.
Thank you. Very wonderful talk, great talk, exciting stuff. So I'm just gonna throw out some suggestion. If you're gonna do experiment, these days, you can actually have machine generate synthetic sound. So they can systematically manipulate visual sound, oral sound, these things. You can do that, you can see that what characteristics of sound.
Oh, totally.
And relate that to brain.
Yes. I'd love to do that. That's one of the reasons I have this database with a variety of acoustic properties, so we can kind of use machine learning to figure out what components are bothersome. The thing, though, that it's difficult with this type of research, I have participants tell me that sound sounded fake. It sounded not real to me, and therefore it didn't bother me as if it was a real sound. So it's a fine kind of line to walk. Yeah.
Any other general questions?
Anything online?
Oh, I don't know.
Any questions from the people?
I do not have any general questions.
Once again, thank you so much, Heather.